Good evening, everyone. Good morning to some of you. Thank you for joining us today. As you can see, we have an interesting topic. Let's start with a prayer. And may I request Jayendra to offer the opening prayer. Jayendra, can you hear me? Can you hear me, Jayendra? Okay, I don't think we can get him to unmute. May I request Michael Aya to offer the opening prayer? Michael Aya, can you hear us?
Can you hear me? Can you hear me?
Yes, we can hear you.
Okay, let's pray then. Thank you, Heavenly Father, for this special moment that we are going to learn about your creation and after creation, what had happened to this wonderful world. We come to know many things through different people. This evening, especially be with Brother Ruwan as he comes out with some information so we can understand more things about where we live and what we are going through and many things, many more other information about this world. Heavenly Father, as these brothers and sisters are focusing on this educational program, let them be with much of knowledge of thee and where we live. We thank you for all the goodness and blessings that thou hast given to each one of us. Bring us closer to your love and care, and finally we may be found in thy eternal kingdom. This is our prayer in Jesus' holy name. Amen.
Thank you very much for the prayer. Just let me remind you to keep your mics as mute and your videos switched off at all times so that we pay maximum attention to the speaker. Also, when you have any questions, please type it in the chat box and I will read it out to the presenter at the end. Today's presenter is Ruwan Wijetunga. I call him Ruwan Aya. He will be presenting on the human microbiome. He has an MPhil in biochemistry. He has been working in the Lakeside lab for almost a bit over 20 years now and has a lot of experience. We constantly discuss about many things and I've learned a lot from him. He's a scholar, but I think an important point to mention here is that he loves studying the Word of God and preaching about the gospel. So without further ado, Ruwan Aya, let me hand it over to you. Let me stop sharing my screen so that you can share yours. Okay, so over to you. Thank you.
Am I ready to go?
Yes, we can hear you. We can see you also. And you can try to share your presentation now.
Okay.
Yes, good to go.
Okay. Okay, hello everyone. It's nice to be able to share a little of what I know with all of you. I have listened to some of the content shared on Youth Inspire. I mean, a lot of variety and color. It's amazing to know how many knowledgeable people we have around with us in our church here in Sri Lanka and also abroad. I mean, this is the age of knowledge and information. I think it's a wonderful idea and a very timely one. And I thank the organizers who came up with this idea to create a platform like this that everybody could be informed. See, microbiology is a very interesting field to work with. I handle microbes on a daily basis. You know, compared to the trillions of highly specialized cells we have in our bodies, you know, a microbe has just one single cell. Can you imagine that? Just one single cell, and look at what a lot of damage that they have caused. You know, the very fact that we are conversing in this manner it's because of one single virus, you know, that got out of control and spread rapidly across the world, you know, due to the globalized nature of our world, due to fast travel. You know, it got around very, very fast. You know, this microbe that we're dealing with is not even considered to be, you know, fully a life form. It's just an RNA strand and some proteins and lipids that are covering it. So these microbes have big potential, and it's very interesting and very important to know a little about it. That's my purpose of today's sharing my content, is to draw your attention to the human microbiome. You know, it sounds like a big word, but it's simple. The human microbiome, if I have to define it, it is the collection of the usually non-pathogenic microorganisms. Pathogenic means disease-causing. It's a collection of non-pathogenic organisms which reside in our body, maybe on the skin or more, maybe in our, you know, various anatomical sites. You know, the collection of these microbes in humans are called the human microbiome. It's a collection of bacteria, viruses, and fungi that live normally within our body and usually does not cause any disease. Right? So these are the different anatomical sites that you can see here on this picture. You know, certain microorganisms can, you know, perform tasks that are useful, right, to humans, many of which their roles are not very clearly understood as yet. It is a very interesting field of and a very active field of research. You know, some may not be friendly to us in some conditions, especially when there's an overgrowth of normal non-pathogenic bacteria, or when your immunity is low, or when you are, you know, taking chemotherapy or radiotherapy. Sometimes imbalances can cause these harmless bacteria, viruses, or microorganisms to be pathogenic, right? So these normal bacteria or microbes are called normal flora or normal microbiota, or maybe known as commensal flora, right? Harmless, helpful, friendly, right? So till recently, a lot of information about the microbiome, the human microbiome, was not known. So the Human Microbiome Project, you know, it took on a study to sequence the genome or the genetic material that we are made up of, that is our DNA, the genes that, you know, we all know of microbes that are part of the human microbiota. They focused especially on microbes that are that normally inhibit the skin, the mouth, the nose, the digestive tract, and the vagina, right? So in the year 2012, they published their results, right? I'll just give you some amazing facts that came out of that research. For example, the study reported a gene catalog of 3.3 million non-repeating genes in the human gut alone, right? Compared to the mere 22,000 genes present in the entire human genome. The gene of a human has 22,000 genes, but look at the collection of bacteria and their genes, 3.3 non-repeating genes in the human body. Let me share a few more interesting facts, amazing facts. The human microbe includes around 100 trillion bacterial cells, 100 trillion, right? The human body has more bacteria than the stars of a Milky Way galaxy. Our Milky Way galaxy has more, the human body has more bacteria than the stars of our Milky Way galaxy. You know, the adult human typically has around 10 trillion human cells, but look at the number of the bacteria in our microbiome, 100 trillion, right? That is 10 times more bacteria than the number of cells that we have in our body, right? So if you line up all microbes in your body, it would go around 2.5 times around the earth. That's an amazing number. That's an amazing number, amazing length, 2.5 times around the earth. And the human microbiome, the entire human microbiome weighs only about 200 grams, only about 200 grams. And all that bacteria you will find in that 200 grams, right? And to tell you frankly, that 200 grams is mostly in your excrement or in your stools, right? Because the gut has the gut has the largest number of bacteria of our human microbiome, right? So the study on the human microbiome started with a man called Anthony van Leeuwenhoek, who lived in the 1680s, and he was a very curious man. Anthony van Leeuwenhoek, he was a clothes merchant. He developed lenses to study the textures and the weaving, and to see whether there are any corrections to be made, any defects in his clothes that he purchased and traded. So he developed these little lenses together, put up little lenses together between plates of aluminium, like you see here, and he did not stop with examining his clothes, but he went on. He was a curious man. He, used to scrape some, cells from his inner cheek, put some of his fecal matter or his stools, take some pond water, and see whether he can identify any minute creatures. And this is what he saw. These are his exact drawings that you see here. The figure A, B is showing that this microbe is moving, right, from point B to point D, right? And this is what he saw. So until this discovery, the microbial world was, entirely, not seen to the human eye. Nobody knew about that any such minute creatures exist. And, in the medieval days, they connected diseases with evil spirits, and things like that, because a cause could not be shown. Right? So after the microscope was developed later by Robert Hooke and people like that, the entire study of microbiology accelerated in a very rapid way, and it is still a very active field of research. And this is a microscope here. It's called the trinocular microscope, a very interesting gadget. Now, first came the monoocular with, one eye. In schools, you remember you close one eye and try to, gaze into the field. Then came the binocular. Right? Now we have the trinocular with another eye here, the electronic eye. With a nice little screen here. and very convenient, even to teach young technicians or pathologists or doctors. A very important gadget, not very expensive. A microscope like this recently about 3.5 lakhs, and a very convenient tool to study, especially for those who are curious. It's a good tool. Even parents, if you want your children to look into the unseen, you can buy them. Maybe instead of a cell phone, you can buy something like this. Okay, just an idea. Right. So the development of the microscope opened up an entirely new field of study. Right? Amazing. And we must remember that the human body is not all riddled with bacteria. There are sites, there are sterile sites in the human body. When I use the word sterile, that means there are no bacteria, viruses, fungi, or their spores, none of them in these places. For example, the blood. The blood is sterile. If you have microorganisms in the blood, it's serious. Okay, then your cerebrospinal fluid, that is the fluid that is within your the spinal cord, your brain, right? That is sterile. If you have organisms in that, the doctor might say that you have meningitis or some kind of infection in there. Then your pleural fluid. Pleural fluids are fluids that surround your lungs. Then your peritoneal fluids, that the fluid that runs in your outside of your abdomen. Then the pericardial fluid, fluid that surrounds your the heart. Then the bone, joint fluids, and all these internal body sites I have listed, like the lymph nodes, brain, heart, liver, spleen. All these are free of microorganisms. If there are microorganisms in these, that's big trouble, right? So these are sites that have no microorganisms at all, right? So the microbiome of humans are limited or restricted by our immune system to certain locations or certain anatomical sites of our body. Okay, now this is a baby. The picture one is a baby in the womb or in the uterus. The uterus is usually an sterile environment. A baby swimming in that amniotic fluid is sterile, no microorganisms. Some research, some studies have suggested that there could be a few microorganisms, but that study is not conclusive. But let us consider that the human embryo or the fetus, the fetal conditions in the womb is sterile. Right? So how did we gain all these microbes in our body? Yes, we call it colonization. Colonization occurs mainly during birth and after birth, right? If you had a normal vaginal birth, then your colonization will be different, because the bacteria that is encoded in the vagina, you get a painting of it when you come out, right? And then it's healthy. It's the mother's microbiome, the vaginal microbiome is specially it's specially designed to give or transfer the mother's microbiome to the child. But if you have a cesarean birth, then it's a little different, because cesarean procedure like here is done in under sterile conditions, right? So, and research studies have found out that babies who are born by normal vaginal birth have lesser chances of developing asthma, and type 1 diabetes. Right? Studies have shown that. So it's all important. Microbiome, the microbiome is very important. By the way, this is my daughter here during cesarean surgeon. This is Dr. Nawaz, long time ago. Okay, now during pregnancy, the placenta provides the fetus with everything it needs to develop, but not microbes. Right? Now you can see the baby born here covered in a type of butter-like covering. It is called the vernix. This vernix contains proteins that prevent common infections. The microbiome is okay. The normal non-pathological organisms are okay for the baby, but this vernix, so this covering, protects the baby from unwanted infections. Uh, and nowadays you don't bathe the baby soon after the baby is born. Uh, I can remember maybe some long time ago they bathed the baby as soon as it was born, but now they allow this vernix or these white stuff, white proteins to be around for a few days so that the baby receives protection from unwanted pathogens coming and causing disease. Okay, so the seeding or the transfer of microbes from the mother to the newborn is influenced by a variety of factors leading to, like I said, labor and the birth process. Then after 2-3 days time, then the parents come, I mean the siblings come to see the grandma, the grandfather, they all bring what? Yes, microbes with them. Right? The microbiome is slowly getting established in this baby. And during breastfeeding, uh, uh, microbes from the mother's breast and milk can enter into the baby and then colonize the gut. So this is how, and then when the mother carries him, then the father carries him, grandfather, grandmother, right? When people, the more the people touch them, the more bacteria, not necessarily harmful, they colonize the newborn baby. Right? All mothers will know what this picture is. This is the first tool that the baby passes. It's called meconium. Unlike feces, it is not smelly or it does not have any microorganisms. It's free of microorganisms. This meconium consists of intestinal epithelial cells, mucus, right, amniotic fluid, bile produced by the destruction of red blood cells in the fetus, and then water. Right, this is odorless. The baby will pass this, or our mothers will know this. The baby will pass this for 1 or 2 days, and then you can see the effect of the microbes that have made it smelly and difficult to deal with. Okay, so these are the areas that the distribution of the microbiome. You can see that mainly it is the gut, the GI tract, the gastrointestinal tract, that has the largest number of friendly bacteria. Then the oral cavity, the skin, airways, the urogenital tract. And this is wrong. The blood is sterile. The blood cannot have. The eye, the conjunctiva can have microorganisms in it, normal microorganisms, but not the blood. This is a mistake here. The 1% should go to the eye side. Right? Okay, this is another picture to just show you where your friendly microbes are. Right? Mainly, where is it? Yes, it's mainly in your gut, right? In the genital tract, and also in your face, mouth, and places like that. On the skin, it is more or less equally spread, especially the places like hands, your underarms, your feet may be more colonized. That's why those places sometimes smell if you don't take care of those places well, clean it. Okay, so this is the variation of the human microbe. Everybody doesn't have the same type of microbe within them. It depends on their lifestyle, right? The host is the human being, the lifestyle, their diet, host pathobiology. If the host is diabetic or has some illness, then his or her human, microbiome may be a little bit different. Then the environment, if you are somebody who is in Nuerelia, you may have a different microbiome. If you are in Colombo, you may have a different microbiome. If you are in another country, cold country, you may have a different microbiome. So depending on the environment that you are in, your microbiome could be different. Right? Then transient community. The transient community members are, the microbes that come and go. Let's say you touch something dirty, your hands are all loaded with those microorganisms, but once you wash them, they go away, right? So your microbiome also is affected by these microorganisms that you come across daily, come and go, right? These microorganisms don't stay continuously. They come and go depending on, what you are doing and things like that. Then your immunity, right? Depending on your immunity, your microbes can change. Then depending on your physiology, your various, speeds of physiology, various types, various effects on physiology, various microbes can get affected by your different physiologies of people. Then the host genotype, right? If you're a male or female or things like that, it can also change, right? Okay, so let's look into the benefits of our microbiome, right? Benefits, and then we'll look into how we can protect our microbiome, how we can support our microbiome so that we can live healthier lives. Okay, this is how it supports our digestion. A healthy microbiome is made up of diverse microorganisms, which include digestion aiding bacteria. See, some food that we eat, like leaves contain a lot of cellulose, and we are not able to digest cellulose. Good cellulose digesters are cows and elephants and people, the animals that are all vegetarian. They have all the enzymes needed to digest cellulose, but we cannot digest cellulose. So the bacteria aids our digestion by digesting these hard to digest food, right? And they break them down so that we are benefited, and we may gain carbohydrates and proteins out of these non-digestible things, but they could be digested by these gut bacteria. So it aids in digestion. That's one thing you have to remember. And another benefit of your microbiome is vitamin synthesis. Thiamine, folic acid, biotin, riboflavin, and pantothenol. These are all vitamin types of vitamin B. And they are plentiful in your diet, but they also can be synthesized by your gut bacteria, right? And it is estimated that a large or a half of the daily vitamin K requirement is also provided by your gut bacteria. Right? So these are very helpful. Once you have maybe an intestinal infection, your doctor might prescribe antibiotics. And what happens is many of these useful bacteria may also get killed and destroyed by the antibiotic. So that's why sometimes your doctor will prescribe a dose of or a course of vitamin B complex to compensate that loss, right? So it's important not to take antibiotics unnecessarily. Take them only under prescription by advice from your doctor. I'll talk about it a little later, if time permits. But these organisms are very important. They help synthesize vitamins that are needed for you. Even if you don't take them orally, or even if they are lacking in your diet, maybe the microbes will help you out, right, during that difficult time. Okay, supports a healthy immune system. Another benefit, a healthy gut microbiome acts as the first line of defense against ingested pathogens. We put a lot of microbes through our mouth daily. Right? We don't eat sterile food. Right? Even when we cook, all microbes do not get destroyed. Right? So we put in a lot of microbes through our body, but in our intestines, what happens is pathogens compete for nutrients, right? And their action will make it difficult for these pathogens to colonize or multiply or cause disease. One or two pathogens do not cause disease. They have to multiply up to a certain infective number so that they can cause disease. But these friendly microbes, what they do is they compete for food. They don't allow the enemy to have food. They eat up all their food so that they starve and die. So that's how important your microbes are. And also it supports bone health. Some studies have found out that diverse groups of bacteria found in a healthy gut microbiome may help to increase the absorption of calcium. Right? So, I mean, we have in our diets, we have enough of calcium, right? We eat a lot of green leaves. If you are non-vegetarian, you get a lot of calcium. Usual person has no lack of calcium in their diet unless your absorption is impaired or weakened by some kind of a condition. We have enough of calcium in our body, right? Okay. Then another benefit supports a healthy weight, right? A small yet significant key to maintaining a healthy weight may be your gut microbiome. Studies have linked out of balance or non-diverse microbes with obesity. If you have out of balance microbiome or gut microbes, studies have linked that with obesity. Right? Conversely, having a balanced microbiome has been linked to being at a healthy weight. And that's interesting. This study, I have read several articles regarding how microbes or a healthy microbiome can help in weight gain, weight control, right? Maybe they may not have, what do you call it, a very prominent effect, but maybe a small effect, right? Then it supports brain health. The gut-brain axis is how our gut speaks with our brain. This communication may be influenced by certain gut microorganisms that release neurotransmitters, right? Leading to the probability that gut microbes, the gut microbiome may impact mood or cognition. Right? Your mood may be dependent on your gut microorganisms, and even cognition or the ability to learn may be affected. These are new studies that have been conducted, and there is evidence to show that there is a relationship between your gut microbe and your brain. Okay, then heart disease and cholesterol. A recent study in 1,500 people found that the gut microbiome played an important role in promoting good cholesterol. Now, there are two types of cholesterol, the bad and the good. The LDL is the bad cholesterol that, that stores cholesterol in your system, and HDL cholesterol is the cholesterol that takes it out of your body. So this promotes, the gut microbe promotes good cholesterol, and some unhealthy certain unhealthy species in the gut microbiome may also contribute to heart disease by producing trimethylamine N oxide, right, which can cause disease. Bacteria within the gut microbiome, and particularly lactobacilli. We have all heard about lactobacilli that are in our yogurts and sometimes milk, may help reduce cholesterol when taken as a probiotic or prebiotic. Okay, we go to the other side. Protects us against diabetes. The studies have found out that a healthy gut microbiome can delay the onset of type 1 diabetes. I'll just go a bit faster since our time is running out. Okay, how can you improve your gut microbiome? Okay, how can you improve your gut microbiome? Stability, resiliency, and diversity, right? About stability and diversity, we can do something. Resiliency is the ability of your gut or gut microbiome to be re-established after it has been disturbed, maybe by antibiotic or maybe by intestinal parasite or infection. Right? Resilience is the ability of your gut microbiome to become re-established. Right? So our daily life choices such as diet, exercise have a profound effect on our microbiome. Right? One of the three diversities that we have the most control over. Right? The stability and resiliency, we have no much control over it, but diversity, yes. Right? You can do things to increase the diversity of good microbes, right? One is eating prebiotic food. This can lead to a diverse microbiome, which is an indicator of good gut health, right? Especially legumes, beans, fruits contain a lot of fiber that can promote these healthy bacteria. What you are doing in eating this food is actually you are feeding your microbiome. You are feeding your gut microbes. They love to eat, they digest these fibers and these the vegetables that I mentioned here, some fruits, right? When you feed them with what they like, they grow well, right? Uh, and the more diverse foods you eat, the more diverse microbiome that you can develop in your gut, right? Especially when you eat, now these foods are called prebiotic foods, right? Now, especially when you eat whole grain meat, whole grain cereals, you can see that this endosperm contains mainly of carbohydrates and some vitamins, but you can see the outer fiber, that is the bran, fiber-filled outer layer, right? Now, it is difficult for us to digest it. What happens is the microbes are very, capable and very efficient in digesting. So this inner part is for you, the outer part is for your microbiome. Right? There are also other benefits like fiber, what fiber can do to your intestines, in stool formation and things like that. But remember that it is important for your gut microbes. They like it. Right? They thrive on the outer layer, the fiber-filled bran. Right? Okay. Then eat foods rich in polyphenols. Polyphenols are, phenolic compounds that are sometimes found in food, like chocolates, berries, olive oil, whole grains, right? They are broken down by the microbiome to stimulate healthy bacterial growth, right? So they also feed your microbiome as well as they feed you, they feed your microbiome. So that's important. Then try a plant-based diet, especially if you are having, you know, digestive issues, and you think that it is, or your doctor thinks that it is your, gut microbes that are causing it. Try going vegetarian, right? Because you eat a lot of sugar and meats, the E. coli that we do not want growing in our intestine, they're like those stuff, the E. coli thrive on sugar and high proteins, but the healthy bacteria, they thrive on vegetarian food, whole grain foods, right? And they also reduce intestinal inflammation and also has a reducing effect of cholesterol levels, right? And then we all know this. Uh, Try to eat as much as you can fermented foods. They are also prebiotic in nature. Fermented foods such as yogurt, sauerkraut, we don't have it here. This is sauerkraut is made out of fermented cabbage. I have never tasted that. I don't know how it even looks like actually. Then kefir, that's a type of milk food, I guess. Right? They all contain healthy bacteria. Now, if you take this drinking yogurt that you find in shops, they have that name, prebiotic bacteria contains prebiotic bacteria. But sometimes the set yogurt you have, I tried culturing, one of those yogurts, but it did not heal any bacteria. I guess they have been destroyed by pasteurization or some process to eliminate those. But the drinking yogurt, but I must warn you that is full of sugar, but it contains healthy bacteria, right? And these are all species of lactobacilli that are used in production of, yogurt, cheese, and fermented food like this, right? So these can reduce the amount of disease-causing species in the gut. If you have a little tummy upset, sometimes when you eat a cup of yogurt or some kind of prebiotic food, it may help. And then how you can improve your microbiome? Breastfeed your infant for at least 6 months. Breastfeeding is very important for the development of the gut microbiome. Children who are breastfed for at least 6 months have more beneficial. This is bifidobacteria. That is the kind of bacteria that we have most in our gut than those who are bottle fed. When the uh, child clings on to the mother's breast and sucks, a lot of bacteria that is around the breast and also in the milk get transferred to the baby, and the baby is protected from intestinal infections that sometimes we often see in bottle-fed children. They always have some kind of a infection going on sometimes, not always. Right? Okay. Take probiotic supplements. Right? I think 5 or 10 years ago, these things started coming up to the market. Probiotics are live bacteria that can help restore the gut to a healthy state after dysbiosis. Dysbiosis is when your gut microbes are disturbed, maybe by infection or maybe by excessive antibiotic use. Right? So what they do is they reseed these probiotics. These are capsules that contain frozen or dried bacteria, harmless bacteria. They reseed your gut, right? They reseed your gut, and sometimes nowadays doctors are reluctant to maybe prescribe antibiotics. Instead of that, they might give you probiotics, right? Just to re-establish or to reseed the your gut that has been disturbed by disease or antibiotics or some other reason, right? Probiotics, you can find this. Take this only under prescription. Any anything that is not advised by a doctor, I don't support that. It is better to always talk to your physician and get the physician's advice before you take any of these. These are harmless, but better to take your doctor's advice. Okay. Then take antibiotics only when necessary. Right? Antibiotics kill many bad and good bacteria in the gut. Antibiotics cannot differentiate from the good and the bad bacteria that live in your intestine or the large intestine. Possibly it can contribute to weight gain and antibiotic resistance. Antibiotic resistance is a huge problem the world is facing. So take antibiotics only when necessary. I know in India it's a huge problem. There is antibiotic resistance to even carbapenems. Carbapenems are kind of the last resort antibiotics. The group of antibiotics. And we see that even I'll show you one slide that we isolated here. These are becoming common. And recently in the Kennedy General Hospital, 5 patients died because the doctors didn't have any antibiotic left to give them, resistant to all. Right? So from childhood, don't run to your doctor for even a slight cold. Sometimes you go to a doctor, the doctor is also tempted to give you something. He may, we all have that experience. Sometimes they, to satisfy the parents, they may give antibiotics, which, may not be really necessary. Maybe you can do it without antibiotics. Do some steaming, some fresh air and some, remedies. Grandma's remedies will do. Sometimes, but antibiotics are life-saving drugs, no doubt about that. But if you use it unnecessarily, you cause difficulties, you cause problems. Even the use of these disinfectants these days can cause antibiotic resistance. Hand washing is fine, but sometimes we use them. Now we see bleaching, the streets, and what happens is bacteria, they grow resistant to these disinfectants, and sometimes we may breed a new generation of microbes due to careless use of disinfectants or even antibiotics. Right? So this is this is called the ABS test we do. You can see Now these small circles here, they are antibiotics. They are discs that are laden with, maybe erythromycin or amoxicillin or things like that. You can see a zone around it. Now this opaque area is bacteria growing. You can see that around this antibiotic disc, there's a zone that the bacteria cannot grow. So what we do is we measure the diameter of this zone and decide whether this organism could be killed by using this antibiotic. Right? This was isolated by a female in a wound in her leg, a chronic wound, and sorry, this is not this is another patient. This shows a lot of sensitivity. Some drugs are resistant. You can see here, right? This is the lady that I'm talking about. This is a normal, not a very resistant organism. You can see there's a little zone inside this, but that is not enough, right? If you give this antibiotic to the patient will not respond to the antibiotic. You can see some have no zone at all. And look at this. This is the lady I was talking about, who had a chronic wound in her leg, and we did a culture, and look what we found out. Resistant to all available antibiotics. And those who know drugs, ciprofloxacin, imipenem, meropenem, cefuroxime, then there is gentamicin, then amikacin, resistant to all, right? So in a case like this, it's difficult. You have to use a combination of very powerful antibiotics to treat them. Combination antibiotic therapy may be used to treat this kind of patient, right? So this is done by misuse of antibiotics. When you keep, attacking microbes with antibiotics, they develop their own weapons, right? They develop their own weapons and they attack the microbe at the antibiotic back, and sometimes they win. And this is a situation that the bacteria has won against antibiotics, right? So even the use of antibiotics against animals like chicken and cattle, it's dangerous because their dung will contain microbes that have been made resistant by the use of antibiotics, right? So even the use of antibiotics in animals also should be controlled and used with care. Otherwise, we cause unnecessary resistance. Now, this kind of organism is a good research organisms. If you can find an antibiotic or some substance, or maybe an Ayurvedic plant or some extract that can, destroy these microbes, well, maybe you can win the Nobel Prize next time. Right, so these are all important for research. These are superbugs, right? This is called a superbug. So we have a few superbugs around here, even in our country, that we have to be careful of, and we have to be mindful not to create these superbugs. Okay, now this is another interesting thing. We have heard about kidney transplants, liver transplants, heart transplants, and this is stools transplant, right? This is the process of transferring, fecal bacteria, in your stool, in your excrement, and other microbes from healthy individuals to another individual. It's called FMT, fecal microbial transplantation. Right? It is very effective against a pathogen called Clostridium difficile. It's an intestinal infection, sometimes difficult to get rid of, right? It may be more, this procedure of stool transplant may be more effective than vancomycin. Vancomycin is a very potent antibiotic. Usually every bacteria, or most of the bacteria are very gram-positive bacteria are sensitive to this organism, but this is more effective than that. Now look at the process. What happens is, this is your stool, and what we do is we mix it with some saline or solution, buffer solution, blend it up, filter it and make sure, you know, all the undigested food particles and things like that. We are not interested of all that. So we filter it so that we only have what? The microbes, right? Only the microbes of someone's feces. And what we do is using the help of an endoscope, you can transmit, or you can what do you call it, using a syringe, you can shoot it into a person's rectum to the anus, right? Pretty interesting, but or sometimes if you don't, maybe you can have it in a capsule form, right? These capsules inside contains frozen stools full of useful microbes. You take it orally and it will get released in the area that is needed, that is large intestine and your rectal area. It will be released so that the lost or the disturbed microorganisms will again get established by this. Right? I can remember one patient. It was a person from Loma Linda who came to visit our laboratory, told that they did it 3 times to one patient, stool transplantation, and on the 3rd time it worked. Right? So sometimes when there's no other option, maybe, you know, taking in somebody else's excrement or stool may be the only option left. Right? Okay, so that is how important your microbiome is, right? Remember, 1/3 of the weight of your stool is all bacteria, friendly bacteria. 1/3 of the weight of your stool is friendly bacteria, right? So better think before you flush. Okay. Now I was wondering how I could. That is about bacteria, and I was wondering how I could draw a biblical, spiritual lesson out of this microbes. I just have 10 minutes. When did God create microbes? You know, the bacteria, the fungi, and the viruses. When did God create them? Yes. The answer is we do not know. Right? Where do microbes fit into the creation account? Were they created along with the rest of the plants and the animals during the first week of creation, or were they created later? You know, these are some questions that, you know, creation microbiologists have been asking, in recent years. The answer to these questions are not clear in scripture, so we cannot create any doctrine out of it. But in my view, microbes had to be there from, you know, the very beginning of life. It seems reasonable to me that microbes could cause disease only after the fall. You know, this is all maybe, okay, because disease came after the fall. You know, some have postulated that microbes were created on a single day of creation, such as day 3. You know, when the plants were made. You know, this is particularly due to the seed-like character of bacteria and fungi, right? But we really do not know. From the beginning, God has made his creation fully mature and complete. That is what we know for sure. From the beginning, God has made his creation full, mature, and complete. Genesis 1:31 says, and God saw everything that he had made, and behold, it was very good, he says. And the evening and the morning were the 6th day. So at the end of the 6th day, before he created Adam and Eve, everything was good, right? But what happened? Though Adam and Eve, through Adam and Eve, God gave humanity a complete, finished, and perfect created world. You know, when God finished creating the world, you couldn't add a single thing to make it more beautiful, or you couldn't take out a single thing to make it more beautiful. Right? You couldn't make it better. It was perfect. That is the kind of gift that God gave humanity through Adam and Eve. You know, but we all know what happened. Sin spoiled it. Right? So through Jesus, the second Adam, God gave us redemption. And sometimes we do not realize that it too is complete, finished, and perfect. Don't try to add anything. You cannot add anything to make redemption better or redemption more complete. Right? Both creation and redemption are God's perfect gifts to humanity. Perfect. When God gives something, it's perfect. They cost nothing to the receiver, but redemption cost a lot to the redeemer. It cost enormous amount to the redeemer, but the receiver, it is totally free. That it costs nothing to the receiver. That is the beauty of redemption. You do not have to do anything but receive it as Adam and Eve received God's perfect creation. Right? Sometimes we don't have a problem with receiving God's creation, right, as a perfect and finished product. But sometimes we find it not easy to accept redemption as complete and finished. Think about it, right? When it comes to our salvation, sometimes, we think maybe we should help out God a little bit. He has done so much for us. Maybe we should give him a helping hand. So maybe I should, impress him by my performance. Those things don't work. Those things make Christianity miserable, right? So that's the thought I wanted to share with you. So it is my prayer that we receive this gift and we receive God's gift of perfect redemption, perfect, complete, and finished, as he said on the cross. Nothing more to be done, but only to be received. Thank you. Thank you, Prabhu. You can take over.
Thanks, Ronaya. There are a few questions, and let me read
Yeah.
them out to you. What is the role of bacteria in relation to COVID-19?
Well, COVID-19 is a virus, and we have won the battle against bacteria. We have, except for a few superbugs, but that too are manageable. But the fight against viruses, we are nowhere close to winning. And bacteria and viruses, they are two different organisms that act very differently. And there's no connection, as far as I know, between your microbiome and COVID-19. No.
What are the harmful effects of continuous hand washing and use of sanitizer on the microbiome?
Okay. In your hand, mostly you have a transient microbiome that it comes and go. I cannot see any bad thing happening by sanitation. Even if you lose some bacteria, you are very quick to gain it back, right? So having your hands clean at this time is important. You have seen sometimes people, sometimes with certain conditions, wash their hands continuously, and you see fungus growing in their hands. Why? Yeah, because the not only the bacteria, the normal bacteria, but also the what we call it, the stuff that is produced through your sweat glands, they are also lost, and your balance of microbes on your hand can get lost. But you must always weigh the risk and the benefit. But right now, it is very important to sanitize our hands. It cannot cause, any danger, as I see.
How can we check our microbiome levels? Is there a particular test that we can do?
Not that I know of. No, I don't think you can check their levels. I don't think so.
In our present world, is it safer to live in an infected or sterile environment?
A sterile environment is not practical at all. Then you have to remain in your mother's womb. Once you are out, it's better to stay away from dangerous organisms. But sometimes it's inevitable that, you get infected. But maintaining a healthy microbiome will, keep you away from getting infected with all these unnecessary organisms. That's that I can give you. Live a healthy life, eat probiotic foods, and do the other health-wise practices that you do.
That's all the time we have for today, Ruwanaya. Thank you very much for taking time and preparing the presentation. And it was certainly very informative and very useful. Let me quickly announce that, let me announce first that we will not be having these sessions 3 times a week from today onwards. Everybody is getting busy and we thought that it might not be practical to run it 3 times a week, but we will run it on a weekly basis. So our speaker for next Sunday, 7th of June, will be Dr. Shivanti, and her topic will be, let not your heart be troubled. So that's something to look forward to. And I'm sure it'll be a very interesting presentation. We will share the links and the poster during the week so that, and we'll remind you of the session. And of course, don't forget to like us on Facebook, Youth Inspire LK. But please, if you have any suggestions, contact us through our social media channels. You have our numbers, give us ideas on how we should continue such programs going into a period where the lockdown doesn't necessarily exist. But we would like to continue these programs and to do something creative. Thank you very much again, Rona, for your time.
Thank you very much. Thank you all for listening. I hope you were blessed.
Yes. Would you mind closing with offering the closing prayer?
Me?
Yes.
Okay. Let's pray. Dear Father, and we thank you for this time that we have. We thank you for the protection you have given us. And we thank thee for your guidance. We thank you for this team that have organized this platform that we could share information about you, about your about current events, about nature. We pray that thou would make everybody understand that as creation was perfect and finished, redemption too is perfect and is finished and is ready to be received by all of us. It is my prayer for everyone. In Jesus' name I pray. Amen.
Thanks again. Thank you everyone for joining us. I wish you all a good night, pleasant day. See you all next Sunday at 7 p.m. Sri Lankan. God bless.
God bless.
God bless. Thank